So this is showing nest weight on the left panel and reproduction, so the total males plus pupa on the right. And the small colonies on the left and big on the right, respectively, on the x-axis. And so if you look at the small colony group, we found no significant difference between the blue and red control and inoculated, respectively, groups. But in the big colonies, we found a significant reduction in total nest weight in the inoculated colonies. And we see the same trend when we look at total reproduction as well. So finally, infection load. This is how many Cryphidia cells we counted in each bee's gut. And so this got interesting. So we might have predicted that given the big colonies having the increased cost of disease, that might be because they have really high infection levels. But this is the opposite of what we found. So we've got the Cryphidia cells, the infection load on the y-axis, and colony size, small and big, on the x. And you can see that the big colonies had significantly less infection than the small colonies, which was kind of puzzling. And we don't know exactly why this is, but we've got some ideas. It could be one that, so we can't screen the dead bees, just a limitation of the method. So the highly infected bees could just be dying off in the big colonies, making the infection load look like it's less than in the small colonies. And it could also be that the bees in the big colonies have less good body condition, which leads to not, like the bee's body condition affects how well the pathogen does, and so they may have less pathogen despite also having increased costs. So wrapping that up, I want to take a beat to talk about the difference between group size and bee density in this experiment. So group size is like how many total bees in the colony, and density is the bees in each given area. And these, while closely connected, could lead to slightly different outcomes. So yeah, to remind you what we did, in the previous experiment that I just explained, we held cavity size constant, but we changed how many bees each colony had. So as we increased colony size, we also increased bee density. And we can't really untangle those things with this experiment. So to try to get at this, I am in the process of a second experiment in which we hold colony size constant and change bee density. We'll change cavity size. So we keep the same 25 bees in each colony, but we reduce the space that they have by 3. And besides that, the design is identical to the last experiment. And I'm still collecting data, so TBD on final results on this. But we find quite different outcomes when we change cavity size, holding group size constant. So if we look at mortality, we've got time on the x-axis and fraction dead on the y-axis, just like in the previous plot. But instead of having only one infected colony, the big infected colonies with increased death, we find both infected colonies have elevated mortality, suggesting that group size in the previous experiment may have been driving the effect. When we look at final colony outcomes, nest weight and total reproduction on the left and right, we see the same general trend. So the blue box plots show the infected colonies, and you'll see that the colonies with the big cavity and the small cavity have pretty close to identical final colony weight and reproduction. And finally, when we look at infection load, while in the previous experiment we had significantly less infection in the big colonies, in this experiment we find almost identical infection load in both the small and big cavity sizes. So all this suggests that it's actually group size, not bee density, driving the increased cost of disease in the previous experiment. So to summarize, we tested how group size affects survival, colony outcomes, and infection load in C. bombi. And we found that increasing colony size leads to increased death, reduced nest size and reproduction, and interestingly, reduced infection load. So, based on this experiment, it looks like increasing group size increases the costs of infectious disease in at least this one primitively eusocial bumblebee species. And although understanding the effects of group size on disease is obviously a huge puzzle, I'm happy to add this one piece. So with that, I would like to thank my lab, the funding agencies, the NSF and USDA, and I'm happy to take any questions. It looks like we have about 2.5 minutes. Yes.
Yeah, interesting. Perhaps I missed it, but is there anything known about cooperative defense against disease in bumblebees?
Yeah, so the question was, is anything known about cooperative defense in bumblebees? And I didn't talk about that much. So bumblebees do definitely remove dead bees from the colony. And I'm also finding, and anecdotally many people have noticed, that they defecate outside the nest. Beyond that, I don't think a ton is known about cooperative defenses in bumblebees, but it hasn't been looked at that closely. Yeah. Yes.
I don't know that much about disease, but how did you decide to infect the relative number of bees in the population as opposed to similar absolute numbers? Like in the larger colony,
Does anyone know what the gong means? Time's up. Okay, I'll talk to you if you want. Cool. Who's next?
Cool. Okay, that's great. I'm a Mac user, so unfortunately, if that didn't work, we'd all be in trouble. But yeah, I'm just going to put a timer here. Thank you very much for coming today. And yeah, my name's Joe Colgan. I'm a group leader at the University of Mainz in Germany. And I guess Canadian is actually one of the nationalities I get most mistaken for over there. So it's nice to be home. Montreal's not a good reference for my accent. But anyway, I'm going to talk to you about colony founding in a superorganism. And this is, first of all, I want to start with acknowledgements, because largely this is work that was the brainchild of Professor Mark Brown at the Royal Holloway, University of London, and Dr. Gillian Lee, based at the Institute of Apicultural Research in Beijing in China. Most of the work that I'll present here today was performed by members of Gillian's group, and this culminated in a paper that was published last year in Nature Communications. But it's a really cool study, so I thought it'd be a fantastic opportunity to come here and talk in Canada. But first of all, starting off with, and I guess social insects can be regarded as what are known as superorganisms. And the concept of a superorganism in terms of social insects was first of all brought about over 100 years ago by the American entomologist William Morton Wheeler. And he noticed from examining insect colonies, he noticed certain analogies that we can see between the specialization of morphologically differentiated morphs, so queens and workers, that we see with specialized cells within the multicellular organism. So drawing connections between queens and workers and what we see in terms of germline and soma. However, not all social insects are superorganisms. And a lot of debates, I guess, have been put forward over the last 100 years. And this, I suppose, served as motivation for a review article that was written a few years ago, which kind of formulated some of the prerequisites for which we can consider superorganisms as being. Amongst these, pre-imaginal caste differentiation, so factors affecting you or genes and pathways being turned on in development lead to you being a certain caste, I guess, like a queen or worker in your adult life. In addition, lifetime worker unmatedness is another classification, and this has been put forward as a major contributing factor in terms of the evolution of superorganisms. So where you have the production of an offspring where the cohort is all unmated, this then leads to a point of no return and an irreversible transition into a superorganism. And in many species, we see that there are certain social but also morphological constraints that are placed on workers to inhibit them from mating. So in terms of social, we have policing, and we also have pheromonal control within social insect colonies. And at the morphological level, we have the loss of ovaries and also the loss of spermatheca, which represents the primary sperm storing organ found in insects. However, in some species which are classified as superorganisms, so they meet these criteria of pre-imaginal caste differentiation and also lifetime worker unmatedness, the retention of the spermatheca in workers always represented a mystery or potential paradox. So species like members of the Vespine wasps, but also the bumblebees, workers containing these structures were always seen as being largely vestigial. So they have a spermatheca, but it was viewed as being non-functional. And this raised a lot of questions of why keep a metabolically or energetically costly organ, which may also contribute to conflict at the social level. So this project had a very simple question to start off with. So is the worker bumblebee spermatheca functional? And here within this experiment, we took some workers, we artificially inseminated them with sperm from unrelated males, and then we tracked colony production and our colony foundation and productivity. Take-home message is yes. So in terms of workers, they once inseminated, they can produce colonies. And here within this figures that we have here on the top row, we have a worker who's indicated in pink who's been inseminated. And on the bottom row, it's a queen that's also been inseminated for comparative purposes. And what we can see here is just these individual pictures, they represent different stages of colony development. So the colony, it progresses, and the main difference that we see between worker and queen-produced colonies is that the worker colony just moves through different stages at a faster rate. However, the worker-produced colony is still able to produce sexuals, so a worker can produce queens. You can get your head around that. It took me a long time to do that too. Then this led then to a follow-up question as well, can workers mate? Like, it's all fine and dandy to kind of show that they can be inseminated and produce offspring, but it raises a question of whether this could naturally occur. They set up an experiment, so a social isolation experiment, because I said earlier that social constraints may inhibit worker mating. So what they did was that they took newly emerged callow workers, they were placed into isolation on their own. Similarly, as a comparative group, they took queens that had just emerged, or gynes, and placed them in social isolation. And then they took individuals that were newly emerged, took them out, tagged them, placed them back into the social colony for 7 days. After 7 days, they entered them into a mating trial with, again, unrelated males. And what they found was that males are attracted to workers, they will mate with them. And indeed, within this experiment, they found that individuals that were kept on their own in social isolation, which is indicated by this yellow band here, had higher mating success. So individuals in isolation, be them workers or be them queens, were able to mate at a higher rate than individuals that were maintained in the colony. Again, providing evidence that social structures contribute to inhibition of worker mating in these species. This then led to asking, well, are there other social factors that influence worker mating? And the first one was looking at, well, queen contact. So if you take out newly emerged workers and you place them in an arena where they're in physical contact with a queen, does this inhibit worker mating? So in this scenario, we have workers that are placed with queens. We have workers that are placed in the same arena, but they're inhibited from interacting with the queen through this wire mesh, so it reduces physical contact. And then lastly, we have these workers, again, that are taken out and placed in social isolation. Similar to before, individuals that are kept either in social isolation or inhibited from physical contact with the queen are able to mate. In contrast, individuals that have physical contact with the queen, they do not mate, and they do not. This implies then that queen contact has a negative effect on worker mating. Similarly, then we wanted to examine what happens when you place them in contact with other workers. And within bumblebee colonies, they naturally remove their queen at a certain point. And this forms then a worker dominance hierarchy. So another question is, well, if you have premature queen loss, do workers actually inhibit each other from mating? And in the scenario, we just have 4 treatments. The first one is taking age-related individuals, so 2 sets of callows and placing them into a container with each other. The second one is placing new callows and then just replacing their nestmates every day. So essentially introducing a subordinate every day. The third treatment is placing a callow with a egg-laying reproductive worker. Thankful the guys in China did this, not me. But yeah, so we have the egg-producing worker. So we've got a dominant individual in the same treatment as our callow subordinate. And then we have individuals kept in social isolation on their own. And what we find is that in all treatments, we have evidence of worker mating, except in the scenario where workers are kept with egg-producing workers. So this implies then that when you have an egg-producing individual, be it a queen or a worker, this inhibits essentially worker mating within your callows. There was other social factors they looked at within the paper, which I won't go through today just because of time, but I wanted to talk about, I suppose, some of the other analyses that were done as part of the paper, and this is kind of more where I became involved. So they were interested in looking at the molecular responses of inseminated queens and workers, so understanding when they receive sperm, do you see similar changes at the transcriptional level? And for this experiment, they took workers and queens, they artificially inseminated them, and then they collected spermatheca, brain, fat body, and ovaries, and compared them to or non-inseminated individuals. What this found is that, and again, I'm just going to focus on the spermatheca for time today, but I'm happy to talk more to people afterwards. What happens is when we do RNA-seq and we look at genome-wide transcription within the spermatheca, what we find is that there's transcriptome-wide differences, so large-scale differences between queens and workers. So just taking a principal component analysis of all these genes, and here what we can see in this PCA is that principal component 1 separates individuals that are queens from essentially individuals that are workers. So this provides, first of all, information that there's fundamental differences at the transcriptome level between your workers and your queens in terms of their spermatheca. However, when we look at principal component 2, and again explains a smaller amount of the variance in our data set, this separates individuals depending on whether they're inseminated or not. So what we find here is baseline differences between workers and queens in terms of their spermatheca, but in terms of insemination, we already start to see patterns of similar responses. We see this though in terms of when we look at pairwise comparison. So if we compare inseminated queens and control queens, and similarly inseminated workers and control workers, what we see is they both elicit similar responses in terms of you get an increase in gene expression within both your workers and your queens in response to insemination. And when we look at the overlap of these differentially expressed genes, we find a considerable proportion. Here we're just highlighting the number of differentially expressed genes, but if you look at higher like gene ontology in terms, you find that similar biological processes are being enriched within these processes. So this provides an indication that queens and workers, that similar pathways are being influenced in response to insemination. The last thing that I wanted to, and slightly take it off a bit detour, just because I want to give a bit more acknowledgement to individuals working in my group in Mainz. What I mentioned was that when we looked at the spermatheca, we found these fundamental differences, and this raised the question of, well, how different are queens and workers overall? So in a lot of social insect research, and I hope I'm not offending anyone here, but a lot of the comparisons are generally confounded by factors such as age, anatomy, or reproductive fitness, or reproductive status, should I say. And bumblebees are a fantastic model because anatomically they're very similar. Like they differ in terms of morphology, but generally they're quite the same. Similarly, you can also control for factors such as age, and now using this experimental data set, we can actually account for reproductive differences as well. What we did was that, and this is work performed by Hongfei Xu, he's a PhD student in Mainz. He was looking at these 4 different tissues that we generated through this study. And here he was just looking at, well, what are the differences between morphs if we want to compare queens and workers. And in tissues that are traditionally, I guess, examined in terms of social insect research, such as your brain due to its importance within behavior, the fat body due to its importance within metabolism and immunity, and the ovaries due to their importance in reproduction, we find very moderate differences in terms of gene expression profiles when we compare queens and workers compared to when we compare the spermatheca. So again, providing greater evidence or more evidence that the spermatheca is kind of a strange organ in the terms of comparing queens and workers. They have it, they may use it in a similar way, but still at a certain level, there are key differences. In contrast with the brain, fat body, and ovaries, there's more moderate differences when we compare queens and workers. In addition, though, and I suppose this is quite interesting, and it builds on other work that's been done, I guess, by certain people in the audience today, I know, in honeybees and pharaoh ants. But we find that cas-biased genes or morph-biased genes are generally more tissue-specific as well. So if we look within the brain, fat body, and ovaries, so these tissues where we find moderate differences, the genes that are expressed in these tissues are generally more specific. So here on the x-axis, we just outline whether a gene is worker, queen, or non-biased, and on the y-axis here we have tissue specificity measured in terms of tau. So a tau value of 0 indicates a gene that's expressed in all tissues. A tau value of 1 means it's tissue restricted. So what it's showing here is that within your brain, your fat body, and your ovaries, generally these genes have higher levels of tissue-specific expression, which largely seems to mean that you could have a transcriptome that's generating a queen and worker in a very similar way, and it's just more localized or fine-scale differences that actually give rise to larger phenotypic differences that we pick up at the organismal level. And as you see with the spermatheca, well, yeah, it seems to be there's just more fundamental differences in terms of transcription which differ between queens and workers. So just to summarize and just give an overview again, so worker mating, it shines lights on the complexity of bumblebees. For me, they've always been kind of viewed as a simple or, a simple organism, but they're highly complex and they surprise us every day. And yeah, the kind of main take-home question of, well, why do they have this strategy? The main take-home at the moment, or the main theory that we're working on, is that the ecology of annual superorganisms, such as bumblebees or wasps probably as well, may select for the retention of worker mating and the ability to express a queen-like gene regulatory network as a backup strategy. So it's quite common within bumblebee colonies for queens to die or queens to be removed. So in case of premature queen death, retention of worker reproductive rights, or reproductive ability, shall we say, should allow for their smash to monarchy. They should allow for them to overtake a colony in the case of that the queen passes prematurely. And lastly then, just at a more general level, the evidence of worker mating suggests that lifetime unmatedness is neither a prerequisite for or necessary expectation of superorganisms. And I guess if you work on certain other social insects such as ants that produce gamma gates, then I suppose you'd probably support this argument as well. But otherwise, I want to say thank you very much. And yeah, if you have any questions, well, actually, it's probably exchange time. I'll probably talk to you at the coffee break, but hopefully you enjoyed the talk.